Molecular hydrogen has been one of my absolute favorite supplements. Why? Because oxidative stress is such a powerful degenerative tool that accelerates not only aging, but our movement towards almost every chronic degenerative disease. So it really is helpful to have a tool that addresses that effectively. So we're going to go deep into this and a lot of the specific details, which you're going to love to hear from Tyler LeBaron, who's probably one of the most brilliant scientists, and he's recently got his Ph.D. in molecular hydrogen. And we're going to go deep in some of the aspects that will help you understand why it's so effective and how to use it properly.
There are genetic mutations where the NRF2 is hyperactivated, and that leads to all sorts of problems. Cancer sometimes can activate the NRF2 really strongly. So what we really are talking about is the regulation of NRF2. When we take molecular hydrogen and we administer it to healthy cells, we don't see changes in the NRF2 level at the protein level. However, if we were to administer a toxin, some other stress, that's when we would see that molecular hydrogen was able to induce the NRF2 or many other proteins, not just the NRF2, and provide a protective effect. And that is, so different than anything else out there. And it has this pre-treatment effect as well. So if, for example, in one study, they took and administered a molecular hydrogen. It was just cell culture, so it's probably only there for like an hour or so in the cell culture. Then after that, they administered a toxin. It's like a common environmental toxin. The exposure caused a decrease in the antioxidant status, so you can see markers of increased oxidative stress. You can see decrease in superoxide dismutase levels, all these important antioxidants, as well as a decrease in your NAD plus to NADH ratio, which having a high ratio is very important. And this stress caused this reduction, okay? However, in the cells that were pre-treated with molecular hydrogen, it prevented those reductions from happening. And it provided a protective effect for 24 hours, even after the hydrogen gas was out of the cell culture. So there was no more molecular hydrogen left. And that's because molecular hydrogen works at this, basically the gene level, epigenetically even, modulating the proteins and the signaling pathways and the phosphorylation cascades. We're not just activating the NRF2, we're regulating its production. So it's not too high, not too low. We are able to provide a protective effect for, say, 24 hours in this case, as opposed to only when it's present, right? That's very amazing to think that a molecule like molecular hydrogen is able to change how the genes express themselves so that it's going to have favorable effects even after it's out of the body.
Welcome, everyone. Dr. Mercola helping you take control of your health, and we're delighted to have Tyler LeBaron back with us again on his journey of continuing to learn more and more about the amazing benefits of molecular hydrogen. And that's a big difference. This is not hydrogen. And I know I was confused about simple hydrogen because that's an hydrogen ion. It's like a pH, it's an acid, but this is different. That's a hydrogen ion. We're talking about molecular hydrogen, which is two hydrogen ions bound together. And most common molecule and smallest molecule in the universe. And it has enormous biological benefits, and it is without a doubt my absolute unmitigated favorite for an antioxidant because it's selective. It doesn't non-discriminately suppress free radicals, which can be highly beneficial. You just want to hit it with a sledgehammer. You want to hit it with a rifle, and it's using your body's own intrinsic biological systems and feedback to understand when you're under this profound oxidative stress. And then it can just activate these pathways, which I'm sure we're going to go into NRF2 and keep proteins and causes your DNA to make the antioxidants themselves, because it's not directly an antioxidant. A lot of people are confused. They think, like I was, and Tyler got me straight, set me straight. It's like you're thinking, the molecular hydrogen is going to bind with the hydroxyl free radical and neutralize it. Well, it happens maybe, but that's such a minute component. It's just, helps you make your own antioxidants. Dozens of them, maybe hundreds. So with all that preface, because you're going to do most of the talking, so I'm so glad you're with us. Thank you for joining us.
Well, thank you. My pleasure. I love talking about hydrogen, so it's a great opportunity.
Yeah, yeah. So why don't you first update us on your journeys? Because I know you're in the process of getting your PhD in molecular hydrogen. And how's that going? Are you getting close?
Yeah, no, so I have finished. I finished last year.
Okay.
And very excited that I was able to be in the laboratory and do a lot of the research. I've been, studying this and looking at it since 2009.
Yeah.
And that's when I started my degree in biochemistry, and then I did a master's degree in exercise and sports conditioning, and did a thesis on using molecular hydrogen. And then I transitioned to doing a PhD as well, and we
Mhm.
did molecular hydrogen research. So
So
yeah,
did you do your thesis and you got your PhD now?
yeah, yep. It's
All
official.
right. Well, congratulations. I didn't realize that.
Okay, yeah,
That's
thank you.
fantastic. Dr. Tyler LeBaron.
Well, yeah.
That's great. That's really great. So, well, I guess that's a big update. And why don't you, give us an update on things, and I'll just figure out
Yeah.
where we go.
I wanted to mention, because this has come up recently again. You said something very important about molecular hydrogen. That's not just the normal hydrogen people think about with the hydrogen ion, which is about pH and acid. But
Mhm.
there's another misunderstanding where people are thinking that in order for something to act as an antioxidant, it has to be negatively charged, and then that's going to neutralize a positive free radical or something.
Mhm.
And that's not really what's going on. And in fact, hydrogen, as you said, yeah, it is a neutral molecule, and it's not donating its electron per se
Mhm.
to neutralize the free radicals. It could scavenge radicals like the hydroxyl radical, which then produces water. So
Mhm.
that's a nice stoichiometry. But really, it's regulating our body's own production of antioxidants. And that's what gives it a unique antioxidant effect. And there's some different things I've seen out there talking about hydrogen, that we should really be looking at H minus or hydride because it has this extra electron. Well, unfortunately, that's not even possible. A hydride is extremely reactive. And to put it in perspective, in order to have a hydride H minus in water, you'd have to have a pH of about 33 or so, because that's based on the pKa,
Jeez.
so to speak. I mean, it's not going to work,
Mhm.
And then anyway, so you don't we just want to we're focusing about molecular hydrogen. All the studies that are out there, there's over 2,000 studies now on hydrogen gas, molecular hydrogen. It's all on molecular hydrogen, not on H minus or H or H plus or all of these things. So it is on H2.
Well, good. And as I mentioned earlier, the primary method, at least as I understand, and certainly correct me if I'm wrong, is that it activates the NRF2 pathway. And are there any specific antioxidants, that endogenous antioxidants your body's producing as a result of exposure to this activation of the pathway, that are more profoundly or more generously produced by your body than others? Like, is it glutathione that's the primary one, or what is the superoxide dismutase? Which one catalyze? Which
So
is the one? And
this
how
is
many of them are there? How many of them are identified at this point?
Well, actually, if you look at the NRF2, so it's a KEAP1 NRF2 pathway. Maybe just to walk through this a little bit for
Yeah.
the audience. In your cell, you have this protein, NRF2, and it's attached to KEAP1, and
Mhm.
they're always attached together. But when you get some oxidative stress or some other stimulator molecule, basically it can cleave off. It causes the cleaving off of the NRF2 and the KEAP1, and the NRF2 can then diffuse into the nucleus, and it binds to the ARE, or the antioxidant response element of the DNA, and then
Mhm.
induces the production basically of all these endogenous antioxidants. It is the phase two, we call them the phase 2 enzymes of the detoxification and antioxidation. There's over 200 of these different
200. Okay.
cytoprotective. Yes.
Wow.
Yeah, it's
Wow.
over 200 of them. So yeah, we typically talk about glutathione as a peptide, a superoxide dismutase, catalase, glutathione peroxidase. But those are just the main, ones we talk about. But there's a whole bunch of other ones, including heme-1 oxygenase, that are activated or can be activated by activation of the NRF2 pathway.
So are there, I mean, that's clearly, it seems to be one of its primary benefits, and its therapeutic uses are almost mind boggling. The number of diseases or indications or conditions, actually, not diseases, can't say that, conditions that it seems to benefit or help or support. So maybe you can highlight some of the most important ones.
Yeah, well, okay. So two things when it comes to the different conditions. You're right. Molecular hydrogen has been shown to be therapeutic in using essentially over 170, probably closer to 200 different animal disease models. And what this means is, so for example, diabetes. There's lots of ways to induce diabetes in an animal model, right? Maybe 10 different ways, let's say. Well, each one has a different there's different factors, different things about it. But using these different models, we can show hydrogen has therapeutic effects. So it is very powerful in all these ways. And like you said, yes, the NRF2 in some cases seem to be extremely important. In fact, by using gene knockout studies or microRNA, or interfering RNA, you can basically blunt the benefits of molecular hydrogen in some specific studies. So the NRF2 is very important to be involved. However, I think we should talk about maybe some advantages of H2 compared to other NRF2 activators like sulforaphane
Right.
or.
Well, I was just going to say that, like from broccoli. I mean, if you can compare the two, because that's the tip. And there's many other
Right.
phytonutrients that activate the NRF2, and broccoli, sulforaphane being one of them, of course.
Yeah, exactly. And it's not people typically want to say, if you have a graph and then this NR2 activator is this much, and this one is this much, and this one is this much, and whichever one is the highest is going to be the absolute best. That's not really the right way to look at it. What we want is optimal levels, and not only optimal levels in the body, but we want optimal levels in the specific cell. Okay.
Mm-hmm.
And so when we take something that's powerful like sulforaphane or other molecules, some of which are toxic, but many toxic molecules also activate the NRF2 pathway. In fact, that is part of the most of these molecules, that do activate the NRF2 pathway are slightly toxic. I mean, that's the whole purpose of them, right?
Tobacco smoke would be an example, wouldn't it?
The which one?
Tobacco smoke.
Yeah, exactly. Yeah, you can see upregulations of this as well. And so most things that are, we like phytochemicals, these phytonutrients, they're slightly toxic for the body, but they end up inducing these favorable effects. Just like when we exercise, all this extra breathing, all this oxygen, we're producing more free radicals, but then in turn, this is going to increase our body's antioxidant status, for example, or increase mitochondrial biogenesis. So when it comes to hydrogen, molecular hydrogen, and comparing this to other NRF2 activators, we need to think about is some cells are already redox. They already have a redox homeostasis. In other words, that the amount of oxidative stress or free radical production is balanced. It's a homeostasis, not truly balanced, but it has the right amount of the oxidative process and reductive process at the same time. That's the homeostasis that we need for optimal cellular health, right? And if molecular hydrogen were to go to that cell, we see this in cell culture studies, for example, when we administer molecular hydrogen in a cell that's already at redox homeostasis, we don't see any increase in the NRF2 or other proteins or things like this. There's nothing like that, and so that's very important. We might see things at the mRNA levels, but not at the protein levels. Now that's important because we don't want to have an issue of having a reductive stress, for example, because if you were to just induce the NRF2 pathway indiscriminately and just keep it going, that would be called reductive stress, and that would be problematic. In fact, there are genetic mutations where the NRF2 is hyperactivated, and that leads to all sorts of problems. Cancer sometimes can activate the NRF2 really strongly, and that can protect it and ensure its immortality. And that leads to a whole bunch of problems. So what we really are talking about is the regulation of NRF2. So like I said, when we take molecular hydrogen, we administer it to healthy cells, we don't see changes in the NRF2 level at the protein level. We might see it in the mRNA level, for example. However, if we were to administer a toxin, say some pesticide or some plastic or some some other stress, hydrogen peroxide or something like this, that's when we would see that molecular hydrogen was able to induce the NRF2 or many other proteins, not just the NRF2, and provide a protective effect. And that is, so different than anything else out there. And when we look at this, and it has this pre-treatment effect as well. So if, for example, in one study, the cell culture study, they took and administered a molecular hydrogen. It was just cell culture, so it's probably only there for like an hour or so in the cell culture. And then after that, they administered a toxin. It's like a common environmental toxin, like a plastic type thing. And anyways, in this case, there was the exposure caused a decrease in the antioxidant status. So you can see markers of increased oxidative stress. You can see decrease in superoxide dismutase levels, all these important antioxidants, as well as a decrease in your NAD plus to NADH ratio, which I know you've talked about a lot before. Having a high ratio is very important. And this stress caused this reduction, okay? However, in the cells that were pre-treated with molecular hydrogen, it prevented those reductions from happening. And it provided a protective effect for 24 hours, even after the hydrogen gas was out of the cell culture. So there was no more molecular hydrogen left. That's because molecular hydrogen works at this, basically the gene level, epigenetically even, modulating the proteins and the signaling pathways and the phosphorylation cascades, so we can have a protective effect for 24 hours. So
that was an in vitro study you described, right?
Yeah, exactly, in cell culture in vitro. We can see other things in animal studies as well, but this just helps hone in on that mechanism that we see. Number one, NRF2 is very important. Number two, we're not just activating the NRF2, we're regulating its production. So it's not too high, not too low. And number three, we are able to provide a protective effect for, say, 24 hours in this case, as opposed to only when it's present, right? That's very amazing to think that a molecule like molecular hydrogen is able to change how the genes express themselves, so that it's not going to, it's going to have favorable effects even after it's out of the body.
Yeah, it's pretty astounding, actually, which is why I'm so fond of it. I think we should review the administration, some of the things you alluded to, in that there's a paradox to the dosing effect, and that if you take too much of it, it's actually highly counterproductive. So I want you to review that, the optimal dosing and the different types of dosing. And I want to know if there's any updates on comparing the tablets to the gas administration, which I know is done in most of the research. Maybe not most, but a significant amount of the research.
Yeah, absolutely. Okay. So when it comes to overdosing, for most people, you're not going to overdose molecular hydrogen that you're able to, and that we're aware of. Because when the studies that are showing the overdosing idea, it's actually that the hydrogen was administered to the animals for 24/7. Okay, so they were continuously exposed to molecular hydrogen, and there was no spiking at all. So in this case, like the cages of the animals, of the rats, they were always had molecular hydrogen, 2% hydrogen gas, always there. And interestingly, initially there was a protective benefit. You could see changes in biomarkers. You could see benefits, and then after a little bit,
first day or so.
I think it was a little bit longer, actually, maybe like the first few days or a week or so. But then it seemed to kind of decrease after that. But when you administer the molecular hydrogen intermittently, that's when it was more effective. And so that's the idea of having an intermittent exposure. So it's probably not possible for somebody to take too much, drink too much hydrogen water, or inhale molecular hydrogen for too much. I mean, some of the studies, people are inhaling hydrogen for 6 to 8 or even 12 hours a day, for example. We see therapeutic effects in this way. So a lot of molecular hydrogen, but not this 24/7 exposure. That seems to be what the problem is. So in terms of the optimal dose, then, so drinking hydrogen water, one thing that we have to keep in mind then is what is the dose that is required in order to induce these favorable changes? Okay. Mm-hmm. If we go back to a cell culture, we need to add so many hydrogen molecules into the cell culture that have to interact in the cell in order for that to activate these pathways. And that concentration, the threshold might be around, 8 to say 10 micromolar. Okay, so you have to get that concentration in your cells. So if you were to drink, say, an 8 ounces or 250 milliliters, hydrogen water or something, well, that's going to have a certain, depending on the concentration, that's then going to be diluted by the rest of your body, and that concentration is going to go down, and hopefully it's going to be at least 10 micromolar or higher. We see some dose-dependent effects from, say, 10 micromolar all the way to 800 micromolar, which is considered saturation. So there doesn't appear to be any problem with having super high doses of hydrogen as long as they're intermittent in this way. But we want to make sure we get that minimum level. So there are probably wrong ways to take hydrogen. And one of them then would be if you have your hydrogen water, and if you just take a few sips and then sit it down, and then a few minutes later, a few more sips, well, you're going to take that hydrogen is going to be the concentration will be reduced by all the by the fluids in the body, so the concentration may never reach that, say, 10 micromolar level.
Well, the other factor too is that the only reason you're able to typically get really high concentrations in the hydrogen water is that these nanobubbles that are created by the tablet technology, and if you if you keep on sipping it, those nanobubbles are going to burst, and there's not going to be virtually any hydrogen gas, molecular hydrogen, in that water.
Okay, yeah, that's another really good point. It's a little bit separate on this, because even in this case, we're just talking about if you have a hydrogen water that's fully dissolved, that's not in nanobubbles, if you're just sipping it, then you're not going to get that concentration of hydrogen. Because when you drink the hydrogen, pretty much all the hydrogen molecules are going to be expelled out of the body via exhalation, most of it, within 60 minutes or so. So if you're kind of sipping it all day, then you're not getting a high enough spike to induce those cell signaling changes.
And does it homogeneously diffuse through the body too, or is it more of a gradient? Whereas if you're swallowing it orally, there's a difference between inhaling it and its ability to penetrate all the different cellular compartments.
Yeah, so it does, because hydrogen is the smallest molecule. It is going to be able to permeate all the cell biomembranes and everything very easily. However, it will follow its concentration gradient, and so it's going to when you drink, it'll go to the stomach and then to the intestines and then onto the liver and then to the systemic circulation, and through Brownian motion and this diffusion gradient, it'll be a passive diffusion, simple diffusion, go into the cells where it's going to induce those effects. So going back to then what's your point about in your case with tablets, if you're choosing to use that, absolutely. The concentration of the hydrogen in the tablets is not just in dissolved form, but it's in this quasi-suspended macro nanobubble form that has a lot of hydrogen density, so to speak, in the water. And so it's not going to be as stable. So you absolutely, in that case, you've got to drink it very, very quickly while it's still cloudy and everything. Otherwise, like you said, you're gonna lose a high percentage of that.
Yeah. So how does that compare? I mean, because I think the percentage is 5 to 9 parts per million, depending on the tablets and such. But the therapeutic dose of regular hydrogen water is like 0.5 parts per million.
Yeah. Okay. So first, on a term, just on a technical term, it is much better that we use, instead of ppm, parts per million, we say milligram per liter.
Okay.
Which in this case is pretty much the same. So 5 ppm is equivalent to 5 milligram per liter, because ppm can mean weight per weight or volume per volume or mole per mole, or all these things. So
chemists would have to make that distinction for sure.
Well, it gets confusing because often when we talk about like hydrogen inhalation, people then talk about 20,000 ppm. And then it's like, really? Because like, what is that 20,000 ppm? How does that equate to, 5 ppm or something?
Right.
One's percentage, the other one is, weight per weight ratio. So milligrams per liter. Yeah, you're right. So So saturation, what we call saturation, is if you were at sea level and you were to put a glass of water in an atmosphere of 100% hydrogen gas, then that gas would dissolve into the water and it would reach an equilibrium. The concentration it would reach would be 1.6, 1.57, more specific, milligram per liter.
Okay.
That's called saturation. Okay. With that. And so most of the hydrogen water that's available on the markets are going to be at best that high, but a lot of them, as you said, about 0.5 milligram per liter, a lot of them are only hitting that level, which according to clinical studies, right now, the evidence would suggest that you really need to be drinking at least that concentration in order to ensure you're going to elicit the therapeutic effect that you want in the first place. So unfortunately, some hydrogen products are not even reaching that threshold. But like with the tablets, like what you're talking about, because you are having so much that's dissolved and a lot of that are in the suspended or quasi, form, then you have a very high volume of molecular hydrogen, which requires you to drink it faster, but you can get these higher doses of molecular hydrogen. So one thing I should probably mention is when it comes to looking at hydrogen products, it's important to one of the considerations is if it has been certified according to IHSA, and I know your tablets have gone through that process, so that we know, for example, that it's really making this much molecular hydrogen. The IHSA is the International Hydrogen Standards Association, which is a group of international researchers that have come to basically give a definition of hydrogen water, look at all the available literature, and to see what is the minimal dose, which we talked about, 0.5 milligram per liter concentration, and then also how to measure molecular hydrogen, because a lot of times people will try to use ORP meters or different things. Mm-hmm. We published a paper showing how this is very inaccurate, and especially if the pH is anything but neutral. So like the tablets, you might measure something like 0.5 milligram per liter, when really it's 5 milligrams per liter because of the issue in the pH. So we really can't use the ORP type meters or other methods. It has to be gas chromatography, and IHSA uses the gold standard gas chromatography, and then does a series of testing for safety and purity and a number of different things in order to be classified as a product that could be recommended and used in clinical studies.
Great. So any indications? One of my favorites is when I travel on a plane, because you're typically flying about 35,000 feet. And for those who don't know, it's about 7 miles up, close, maybe 6 or 7 miles. And that's pretty high. It's much higher than Mount Everest. So the protection of the atmosphere is gone, and the result, there's ionizing radiation from space that come through, like gamma rays. So that can really do havoc on your cellular structure. So I think it's really important to do, take molecular hydrogen. And I think the base strategy is that, just like you said, you cannot take this thing continuously. This is not something you want to take through 3, 4, 5 times a day. You want to take it once a day, maybe twice a day, unless you're traveling. So my approach that I've evolved to is to take it before I take off, because the results, it takes a while, maybe an hour, and you can speak to this, for those ARE enzymes to be activated, or antioxidants response elements to be in the DNA to be activated and start producing these endogenous antioxidants. So you want to have it before the exposure, and then I take it every hour when I'm in the air, just a tablet, and put it in water. I learned this from you too. You want it, ideally, the best amount is 16 ounces of water. That's a big chunk. You're going to get a significant benefit if you put 8 ounces of water, but it should not be cold, because it's going to take a lot longer to dissolve that tablet. And then you got to be holding this, looking at the cup to see if the tablet is floated to the surface yet. So which normally takes a minute and a half at room temperature. It could take 3, 4, 5 minutes, depending on how cold the water is, if you get ice cubes. And not to use sparkling water, because the carbon dioxide in that will inhibit lessen the concentration of hydrogen, the gas that you can have in the water. So that's my take on it. And I just learned recently, I don't know how I missed this, but we actually sell these tablets in little Alka-Seltzer foil tablet packs. So it makes it ultra convenient to travel with them. You just take it out of your pocket, rip it open, it's totally fresh. Because one of the problems with these tablets is that they're very hygroscopic, which means they attract water really easily. And when they are exposed to water before you use them, the benefit is going to be diminished. So you want to keep them as pristine as possible until you use them. So then those packets let you do that.
Yeah, that's awesome. Well, just a couple of thoughts. Okay. First off, I think it's probably okay if somebody wanted to take molecular hydrogen 3 or 4 times a day. The issue would be when you're taking them. So I would say, because we just talked about the pharmacokinetics a little bit, that it takes about an hour for all the hydrogen gas to leave. Then maybe it's better. This is just a maybe. We actually have no research on this. This is just
you would know.
Yeah, this is just my idea on this. Okay. So because we believe it's important to have this spiking effect, then maybe it makes sense that if you're going to have hydrogen water, you would actually not want to have it for, say, you want to have like at least an hour break, basically. Yeah. That way you go back to baseline. The hydrogen levels goes back to baseline so you can spike it again. Okay. So that means, unless you're trying to restrict water intake or something, but you could feasibly take several times, quite a number of times throughout the day, as long as you're having maybe, and maybe you should try to say 3-hour spaces in between or something like that. Now,
at least twice a day. So that actually gives good support for the protocol I advise, which is every hour while you're flying. That would make sense.
Yes. You could do it that way. And then, but maybe you could argue, well, but is an hour enough for it to go, for everything to be
too low? Okay.
Right. So you could wait a little bit longer. Now, it's not actually so important that you take it right before or something, because the activity, the benefits of molecular hydrogen occur not necessarily when the hydrogen molecule is in the cell at that time. Like that study I talked about earlier, you had all these therapeutic cytoprotective benefits long after the hydrogen was already dissipated. So it's like, as long as you've been taking hydrogen, and I would say based upon the animal studies, for about 3 days, then that 3-day pre-treatment is going to make it so that by the time you get exposed to radiation,
brilliant. Yeah,
then you're already going to have that level of protection. Now, I don't think that if you took the hydrogen every hour or every, say, 30 minutes, that you're going to negate the benefits. I don't think that actually would happen, because you're still getting spikes, even though you're not going all the way back to baseline. So there's probably not necessarily a wrong way to do it, but there might be some ways to optimize it and make it better. And I would just say, make sure you're taking the hydrogen water, say, a couple of days before you're going to get on, be exposed to something. And then, of course, before. And then, you can do it. You can do it after also. But I tend to believe that a pre-treatment of molecular hydrogen is going to be more effective than a post-treatment, for example.
Okay, so depending on your circumstances, if you're convinced of this and you're taking molecular hydrogen every day, fine. It's probably closer to ideal. If you're not, and you're trying to conserve and just take it for incidents of high oxidative stress, flying would be one, X-ray exposure would be another, especially CAT scan, which is like 200 times more intense than a regular chest X-ray. Then you want to take it for at least 3 days. At least 3 days before the exposure, so your cells are sort of saturated and primed or ready. It's like they're warmed up and ready to make what they need to. So does that summarize what you were just saying?
Yeah, pretty much. It's probably good recommendations. Of course, these things may change as we understand more of the clinical evidence. In the cell culture studies, I would say that we need at least a half an hour or so. If you just look at the expression of G protein coupled receptors of change from gene expression, and then you make the mRNA, and then you go to the ribosomes, make the proteins. All this stuff takes time, and so around a half an hour may be how much time is needed to start exerting some effects, right? So 30 minutes pre-treatment before something might be important consideration, but again, you don't necessarily have to have a pre-treatment. A simultaneous treatment can be effective. For radiation is something we test a lot. In fact, with my Ph.D., we used a lot of irradiation of the myocardium, for example, and looked at the protective effects of hydrogen water.
What type of radiation did you
Gamma radiation.
Gamma radiation. Yeah, that's the type you get up at 35,000 feet. Yeah. Not typically at sea level, but it is a very potent ionizing stress. There's no questions. Probably one of the toughest. And I think that's the one from nuclear fallout too, if I'm not mistaken, isn't it?
Yeah, I think so. Yeah, because if you're having a particle decay in alpha, we have to look at that. But there's a number of forms, and they all can all be toxic. In fact, non-ionizing radiation can
absolutely.
So it's very interesting when we look at some of the nuances in the biological systems.
I wrote a whole book on that about non-ionizing. It's called EMF.
Yes,
and interesting because we think. I mean, it's speculated. There's a lot of theories on it. I don't think any of it's proven yet. But the current one of the current popular theories advanced by Martin Blank is not Martin Blank, Martin Paul. Sorry, Martin Blank is another EMF researcher. He recently passed. But Martin Paul is that the exposure to the non-ionizing EMFs causes calcium to go extracellular, intracellular, which causes a increase or an influx of the calcium influx causes an increase in superoxide and
the nitric oxide,
and you form peroxide nitrite, which is actually a pretty pernicious molecule because it lasts about 1,000 times longer than the hydroxyl free radical. So I think collectively it may be more pernicious than hydroxyl, certainly not as acutely damaging as hydroxyl, but long term, I think it can travel between cells. I mean, hydroxyl can only travel like a very small distance, like not even the length of a protein.
Yeah,
it's got a billionth of a second half-life. So
right. Yeah, the peroxide nitrite is absolutely one of the most pernicious molecules, and you're right, because it can be protonated too to not form a nitrous, but peroxynitrous acid, and then it's going to make it be able to diffuse easier as well. And one of the decompositions of it is it can create hydroxyl radicals also. So it really is something very toxic, and so I think that's a really good segue, though, into how molecular hydrogen might help with the peroxynitrite.
Definitely want you to do it because it's my supposition. Ideally, this is like in any biology, is the best approach is prevention. So minimize your exposure. But for whatever reason, there are so many people who just, for whatever reason, are unable to limit their exposure. There's a lot of good reasons for it, but that's not the best. The best is to limit it. But if you can't, then you may want to consider taking molecular hydrogen on a long-term basis because it's going to help your body increase the endogenous and antioxidants to minimize the damage from peroxide nitrite and other oxidative stressors. So tell us how it works for peroxide nitrite.
Yeah. Okay. So one of the first studies, actually, the Nature Medicine publication back in
the 2007.
Yeah. Yeah. Well, in addition to showing how hydrogen could act as a therapeutic, antioxidant, one of the things that it found, in addition to its ability to reduce the hydroxyl radicals, was also its ability to reduce peroxynitrite. So there's one way right there how molecular hydrogen can help is we see reductions of peroxynitrite levels, and we also see reductions like in animals and tissue samples of nitrosotyrosine levels, which is a marker of the peroxynitrite as well. Now, you mentioned earlier that, for example, if you get the calcium signals, and that can induce a nitric oxide and activate various NOx enzymes to increase superoxide production, and then you have superoxide and nitric oxide, and they react instantaneously. I mean,
much faster than superoxide dismutase. No comparison.
Yeah,
orders of magnitude faster.
They are, the only thing that limits how fast they react is the rate of diffusion, basically. So, and superoxide dismutase is a very fast, one of the faster enzymes. So you can imagine, essentially it means that if they come in contact with each other, they will form, peroxynitrite. Right. So what we need to do then is, you mentioned this idea of prevention. Well, if we could somehow decrease this excessive production of superoxide or an excessive production of nitric oxide, then we could essentially prevent peroxynitrite formation. And that's exactly what molecular hydrogen does. And this is really fascinating because if you took other antioxidants and you put them in in the presence of, say, nitric oxide or superoxide, you could also scavenge. You could reduce these. You could scavenge them. You could, donate your electron and then neutralize these, basically. Well, that can be good, but can also be bad, because our body makes and specifically uses things like superoxide to increase mitochondrial biogenesis, or nitric oxide, of course, for vasodilation. I mean, that's one of the most important molecules that there is for our immune system, for every, all so many parts and functions of our organs and cells. So we don't want to just neutralize all of these. Well, again, hydrogen being selective, if you put molecular hydrogen in the presence of superoxide or nitric oxide, there would not be a reaction. They don't have the strong enough oxidizing power to for hydrogen to react with these molecules. And so we don't have to worry about that happening. But then the question is, then how does hydrogen help with the superoxide and the nitric oxide when their levels are in excess production? And that goes to this signal modulating effects. So as I mentioned, with superoxide, typically that's from this NADPH oxidase or NOx enzymes that can become hyperactivated. And molecular hydrogen has this ability to essentially downregulate this NOx enzyme. And so you end up producing less superoxide in the first place. So if you have less superoxide, then you're going to make less peroxynitrite. And then on the other side, when you have nitric oxide production, you have three main isozymes or enzymes. You have the neuronal nitric oxide synthase, endothelial nitric oxide synthase, and the inducible nitric oxide synthase. And eNOS, of course, that's in the endothelial cells. So typically that's good. You want more of that. You kind of lose that as you get older. Interestingly enough, side note, molecular hydrogen can actually improve eNOS.
That's great.
So we actually can have better blood perfusion and things in this way. But the iNOS, specifically from macrophages, can be problematic. And hydrogen has this ability to downregulate the activity of iNOS of making this excessive nitric oxide production. So now you're decreasing superoxide and nitric oxide levels, and consequently you get less peroxynitrite.
That's great. So that was really, really helpful. Thank you for sharing that. So, and that's what we believe is one of the primary mechanisms of how non-ionizing radiation works. But then we've got ionizing radiation, and you have biological free radical production. And from my understanding, and as one of the most pernicious sources of biological free radical production is an excess of omega-6 fat, specifically linoleic acid. And so that's the substrate for it. And it's interesting. I was listening to a podcast where they reviewed the data on this, and they found that even for ionizing radiation, that the animals that had much lower tissue levels of linoleic acid had far less damage than the animals that had higher levels when exposed to equal amounts of ionizing radiation. So even though they didn't do a study for non-ionizing radiation, it just made perfect sense that lower linoleic acid levels are going to produce less free radicals when you're exposed to these oxidative stressors. So that, I've become less, I've been in a course for the last 3, 4, maybe 5 years of lowering my linoleic acid levels. So they're finally getting down to low levels. So if you can get those seed oils out of your diet in every way, shape, or form, especially in processed foods and restaurant foods, then you're not going to have a lot in your tissues. And when you're exposed to these oxidative stressors, you're not going to generate those free radicals, which ultimately cause the damage. So that's another way that you can preventively, but especially when you're using molecular hydrogen, that's like a massively effective 1-2 punch.
Well, and this lipid peroxidation that's going to occur with the omega-6 fatty acids, I mean, they all these omega-6 fatty acids, they are polyunsaturated fatty acids. They have double bonds, so they have areas where they can actually be oxidized.
Yeah.
And so that's just a fundamental property of them, and then that can induce this propagation cascade, causing more and more. And then when you start destroying the cell membrane, or that cascade is going to enter into the down into the
mitochondria, the nucleus.
Exactly. Yeah. Down to the mitochondrial nucleus. And then, then you start, causing release of cytochrome C, for example, which induces apoptosis, cell suicide, and so on. So molecular hydrogen, it's also lipid soluble. Okay, so it's
Ah,
actually more lipid soluble than it is water soluble. So
really? I did not
yeah,
know that. I thought it was just neutral and didn't have a preference.
yeah, it has a tendency of wanting to be more lipid soluble. So it's several times, at least 3 times more soluble in lipids, for example, than it would be in water. And so you'll actually have more molecular hydrogen in the lipid membrane. And there was a study done by Dr. Ota, who I've done an interview before. He's one of our MHI advisors. But his study.
That's Molecular Hydrogen Institute for which you founded.
Yeah, molecularhydrogeninstitute.com. Yeah, .org now. Yeah, .org.
Yeah.
But anyway, in his study, and it's very interesting. But this was a in vitro cell-free study, basically taking basically a cell membrane type idea and administering a small percent of molecular hydrogen, so it could equate to what it is physiologically.
Mm-hmm.
And when you don't have the hydrogen present, then you get this auto-oxidation. Okay, that's just, oxygen comes, it causes oxidation, you get this propagation. What ends up happening is you form these byproducts, lipid peroxide, byproducts, like 4-hydroxynonenal, and then this sequesters various, other complexes, which prevents activation of AKT, different protein kinases, and then eventually that can cause problems. Okay, because you're suppressing these activities. Now, when you administered molecular hydrogen, there was less production of these lipid peroxide end products, and consequently, there was less of the inhibition of these molecules. So these molecules, these protein kinases, for example, they in turn end up activating things like PGC1 alpha, which is a stimulus for mitochondrial biogenesis, and then eventually fibroblast growth factor 21, which is a hepatic hormone, which is important for energy expenditure and weight loss. And so things like caloric restriction and some of these benefits come in with molecular hydrogen, and it all came from molecular hydrogen protecting the cell membrane basically from this auto oxidation.
That's tremendous. I didn't realize it was so beneficial for that, but it makes perfect sense. It just never occurred to me that would be another useful strategy. So as long as we're expanding on this, those were 2 really profoundly important fundamental biological challenges because with oxidation, because why is it so important? Because so many experts now believe that one of the ways that aging is accelerated is through oxidative stress. So we really need to have a good handle on this, and you got to be careful. And you just can't be you could, but we don't recommend indiscriminately swallowing these antioxidants. It's so far superior to allow your body to do it in its own wisdom and based on the feedback it has from the environment with oxidative stressors.
Well, and there's clinical data also that taking these high doses of synthetic antioxidants, they actually increase mortality. I mean, you end up dying faster, right? But these are the early studies. Yeah, like they found that smokers, for example, who ate more carrots, or something, they're like, hey, they're living longer. So they were recommending, hey, smokers, you should make sure you get a lot of vitamin A and beta carotene. And they're like, hey, we should do a study on this to actually make sure that these recommendations are sound. And they were taking, different studies, and sometimes high doses, you call them synthetic. It doesn't really matter. The point is, these are reducing agents, reducing molecules that have the ability to neutralize free radicals. And they had to stop the studies because people who were taking the antioxidants were dying and getting cancer faster than those on the placebo. And in fact, the recommendation is like, be careful about taking these, especially if you smoke. So
yeah, there you go. So it exactly perfectly illustrates what I just said. That's great. So it occurred to me too, there's another area that I became recently aware of reductive stress, which is an interesting concept because most people don't know about it, but it's when you have an excess of these omega-6 fats, specifically linoleic acid, that it perturbs the electron transport chain. So you get backward flow, and it actually increases reductive stress, which is a challenge.
Yeah, that's interesting. Yeah, so there's probably 2 different ways of looking at a reductive stress in this case. So yeah, when you're looking at the mitochondria, I mean, So at the university I teach, of course, bioenergetics, as exercise nutrition for my master's class students that I'm teaching, and we kind of go through some of these pathways in detail. But yeah, if your mitochondria is not functioning correctly, then if you get back up the electron transport chain, then what happens is just those electrons, instead of going to oxygen as a final electron acceptor to form water, you end up donated it to oxygen prematurely or other molecules, and that causes more free radicals and can be problematic in this case. And then a poor functioning mitochondria, if you're not able to regenerate your NAD plus levels, because that's so important for our overall health, then you end up having more and more of NADH, and this leads to more and more of your NADPH, which and then because you have NADP and NADPH and NAD plus. Anyway, you can start having this type of reductive stress that can be problematic from a metabolic perspective. But then there's this other idea of a reductive stress in terms of having too many, like I mentioned earlier, this upregulation of the NRF2 pathway, for example, right, like happens with cancer or some genetic issues. Or just taking high levels of antioxidants. That's a reductive stress. And so you're absolutely right. With aging, it's and diseases, it's not so much an oxidative stress. We focus on that, but it's really a dysregulation. It's a redox dysregulation. And in fact, we've seen in some of these aging cells, you can have a dysregulation of redox within the exact same cell. So, for example, in the one compartment, let's say the ribosomes, which is responsible for folding proteins. And so you need to have some oxidative power in order to fold proteins correctly. But as cells get older, they may lose that ability. And if you don't fold your proteins correctly, then they're not going to function correctly, because the function of a protein dictates, the structure of the protein dictates its function, right? So this is one problem. And then in the same cell, in the cytosol, let's say, you can have too much of a oxidative stress going on. So you have reductive stress going on in the ER, the endoplasmic reticulum, and an oxidative stress going on in the cytosol. And different compartments can be, all have different issues going on. And so again, just taking an antioxidant or taking just sulforaphane or just taking whatever, that might help one compartment, but it might exacerbate the other. So what we really want is a regulator, a redox regulator, and that's kind of like what molecular hydrogen seems to do.
It is what it is. That's a perfect description of it.
Yeah, the adaptogenic, redox regulator, basically.
Is that a term you came up with, adaptogenic redox regulator?
I did, actually. I published a paper. Yeah, I called it a mitohormetic redox adaptogenic regulator.
I love it. That's great. That's great. You coined a new term in scientific jargon. Congratulations. So one of the other things I admire about your is commitment and passion about exercise. That passion we both share. You're significantly better at implementing it than I've been. You won the state championship in Utah for wrestling when you were in high school, I believe. Yeah. And ran a sub 230 marathon, and you deadlifted over 500 pounds. So you're an extraordinary physical specimen. So I'm wondering if you could maybe touch on the use of molecular hydrogen for exercise performance, and some of the benefits there.
Yeah. So again, we actually published a paper on this in the Canadian Journal of Physiology and Pharmacology a few years ago. But the studies are, there are quite a number of them showing that we can, have improvements in, say, our endurance levels, or I kind of want to go through some of these a little bit. I just reminded there was just an article published as well, a systematic review meta-analysis on the benefits of hydrogen for exercise, and it was just recently published this year, actually. So and again, meta-analysis are some of the strongest evidence that science has to offer, and it shows its favorable effect.
If the articles it's evaluating are valid, because they use that to totally screw with us in the COVID narrative, and they pick the wrong studies to do a meta-analysis with.
Yeah, that's one of the problems with meta-analysis in general is typically it's only valid if we know the authors have a level of expertise in this area. Yeah. But okay, so some of the benefits of with hydrogen with exercise, for example. So first, it seems to be, if it's going to help you, it's probably going to help you as you push yourself harder. So the further away you are from homeostasis, the more you're likely to see an effect in terms of helping the perceived exertion, or helping with your blood flow, for example, or helping to reduce fatigue. And so some of the studies, like one of the earliest ones, found a ability to prevent fatigue during a maximal isokinetic knee extension exercise. So in this study, basically, isokinetic just means same speed, and so you're on a machine and you're just doing these leg extensions. You have to do, I think they did 50 of them in a row, and you just do as hard as you can. And then the group that took the molecular hydrogen was able to maintain a higher force output during those 50 maximal isokinetic knee extensions. And then also they looked at exercising at around 70% your VO2 max, which is about close to 70% of your max heart rate. And those who were doing this, they were able to exercise longer, like a longer time to exhaustion, for example, but also had lower levels of lactate. So lactate, there's a lot of misunderstanding about, lactate. We heard like lactic acid, causes the burn or something. None of that is true. Lactic acid is actually lactic acid itself is not even produced in the body. The only lactate, the molecule lactate, is, in fact, I have one of my exercise phys textbooks. It still has lactic acid in there, and it says that lactic acid is made, and then it disassociates into lactate and the hydrogen ion, which again, it's not true. In fact,
tell us. I thought it was the pyruvate metabolism.
Yeah. So what happens is pyruvate is the end of the you take glucose and you make pyruvate. What happens is lactate dehydrogenase, the enzyme, then takes pyruvate and adds 2 electrons and a hydrogen ion, which is the acid, adds a hydrogen ion to the pyruvate, and that forms lactate. So the molecule lactic acid is never produced. Okay, but I don't want to go too far in the weeds. I'll go really off on a topic, but I want to talk about this lactate just real quick. The reason why we produce lactate is because pyruvate normally will go to the mitochondria to do this PDH complex, and then it's oxidized into acetyl-CoA. It goes anyway. Then you end up making a bunch of ATP or energy in the mitochondria with using oxygen.
Right, aerobic respiration. Exactly.
Right. Okay. So if your mitochondria are unable to keep up with the demand, with the ATP demand, with the amount of exercise, so it's not able to make enough ATP using the mitochondria, using this pathway, because maybe you don't have enough mitochondria, maybe you're not getting enough oxygen there. It's just taking too long, and
or the exercise is too hard.
Or you're exercising too hard, but that's because your body's got to get better, right? So what happens then is you start producing lactate. So lactate is this byproduct. So because the production of lactate is what allows you to continue exercising. It's what allows you to continue, going at that high intensity, and you can make lactate instead of having a buildup of pyruvate. It's actually buildup of NADH, which is the issue. Okay, now in the study then, when they found that you have a reduction of lactate, what that means is that, one interpretation is that the molecular hydrogen may have improved the function of the mitochondria. And we see this in other studies where we actually see increased energy production, so increased ATP production in the mitochondria. Now, if we're getting our ATP production from the mitochondria using the aerobic respiration, then we don't have to go through the anaerobic pathway of making lactate. So that's kind of the important area of why we're seeing lactate decrease, is because we're able to use the mitochondria to make ATP, and now we're going to exercise better, longer, and have less fatigue, especially the perceived exertion, in our brains as well. And then there's also maybe other explanations in terms of lactate clearance and accelerating the Cori cycle and different things. But the mitochondrial bioenergetics are probably a major target of molecular hydrogen.
Excellent. Well, thank you for expanding on that. So mucho benefits when you're exercising. It's a really good idea if you want to optimize and maximize your investment in time and effort and energy. If you're going to put in the work, you may as well optimize it with some molecular hydrogen.
And it's protective because, as we said, when you exercise, you do breathe a lot more. And that's going to make more free radicals. And a lot of those free radicals are going to be very good for your body because it's going to force you to make more antioxidants. It's going to increase mitochondrial biogenesis and all this stuff. But you're still causing damage. You're still damaging DNA. You're still doing things. So molecular hydrogen, the idea here is that you can negate or reduce the damaging effects of exercise while not inhibiting the benefits of exercise, and in fact, maybe even potentiating the benefits of exercise. And so this is one of the ideas that hydrogen, in some ways, can act as an exercise mimetic, not in the true sense, maybe a pseudomimetic, because it can activate some of the same pathways, some of the same metabolic pathways that exercise does. And in this case, it can maybe really potentiate those benefits of exercise. And again, to compare that to conventional antioxidants, especially in animal studies, we see this, that taking high dose antioxidants can negate the benefits of exercise training. So normally with exercise, you have like improved insulin sensitivity, your glucose levels go down, you have better antioxidant status. Taking high levels of synthetic antioxidants can completely negate those benefits of exercise. So again, hydrogen is superior because it doesn't do that.
Mm-hmm. Does it obliterate the external antioxidants that you would swallow? Is it somewhat similar mechanism as cold water immersion or cold thermogenesis?
Well, I guess the end result would be the same, that conventional antioxidants and
different mechanism.
Cold immersion. Yeah, different mechanism. But there, of course, there's going to be some interplay because free radicals are always at play,
Mm-hmm.
But there's also things like the chaperone proteins, the heat shock proteins,
Sure.
And different things that are in clearing of like calcium, inorganic phosphate, and things in the
I
muscle.
love heat shock proteins. Most of my heavy workouts are followed by a near infrared sauna. I get up to like 180 or so, and my body temperature is like 102 for only 20 minutes. And that's one of the I love activating heat shock proteins because, as you mentioned earlier, those protein unfolding is a huge problem. If your proteins, you can have the protein, but if it's misfolded, it's not going to work.
Yeah. Well, and molecular hydrogen also induces the heat shock protein response.
Oh, I didn't know that.
Yeah, so some of my colleagues in Japan, they did several studies. We actually published one recently, but we see that yes, molecular hydrogen is able to indeed induce MTUPR, the mitochondrial unfolded protein response, and then this is important because then this could result later in rejuvenation of the mitochondria, for example. Mm-hmm. So there's lots of heat shock proteins, but hydrogen is involved in this, and then it induces later an upregulation of collagen biosynthesis as well as some of these same pathways.
That's terrific. That's great. So I was thinking as we were elucidating the mechanism with the omega-6 fat linoleic acid, which I view as probably one of the most pernicious contributors to chronic degenerative diseases. So heart disease, cancer, diabetes, obesity. So if you're mitigating, and it's not because it's a direct toxin by itself, but as you mentioned earlier, it's the byproducts, the ALEs that's referred to, advanced lipoxidation end products, which is similar to AGEs from carbohydrates, advanced glycation end products. But these ALEs, or even oxalams, more specifically, oxidative linoleic acid metabolites, the metabolites, and there's hundreds of them. 4-HNE is just one, but there's hundreds of them that contribute to their independent free radicals that damage the cellular tissue. So if you can lower that production of those free radicals to begin with, you're probably radically contributing to almost every chronic degenerative disease.
Yeah, that's why we can explain that molecular hydrogen can have such a wide, diverse benefits in so many different disease models, because essentially all of them have this root cause of an excessive amount of oxidative stress or redox dysregulation we talked about, or this inflammation. And hydrogen able to regulate these pathways is going to help at the root cause, as opposed to a certain drug that's going to target a very specific receptor, go into this specific organ. It's going to one target, one organ and one effect type idea. And that's just not how molecular hydrogen works. It's able to work on the oxidative pathways, on the mitochondria, on autophagy. I mean, there's so many areas where molecular hydrogen works And so it makes sense that it should be able to help with so many different conditions.
Yes, indeed. Well, I love it. As I said earlier, it's my favorite antioxidants because it's selective. It's not like any other antioxidant, and really is really one of my absolute favorite supplements, and I embrace widely. So what's on the horizon for you? You got any exciting research that you're working on, or any theories that you're seeking to prove?
Yeah, one of the well, there's a number of things. Okay, so maybe we really like to understand still the mechanism of hydrogen in terms of what is it actually exactly binding to,
Mm.
what's a primary target, and then this is going to induce this and so on. So that's still being uncovered right now. There is a brand new article that came out that could be a kind of a groundbreaking article. We need some more research to validate it, but hydrogen, it's kind of a complicated thing, but I'll make it very simple though. But this paper just came out, and we have the hemoglobin that can get degraded and to form different. It forms different things when it's during its degradation process, and hydrogen can interact with some of these and actually form this association with some of the iron complexes in this case, and this ends up acting maybe as a catalyst for which hydrogen can neutralize free radicals and prevent oxidative stress, and it can be attached to these proteins in the body that circulates all throughout the body, so it can be brought through the brain, through all the organs and everywhere else, and could help mediate a lot of the effects that we're seeing with molecular hydrogen. So that's a very interesting study. More data needs to be done, but it's pretty neat that we're actually seeing how hydrogen is able to maybe bind to some of these novel proteins in the body. So
Yeah.
that's one area. And then another interesting thing is some people seem to be much more sensitive to the benefits of hydrogen than other, and this is just kind of anecdotally, but even when we look at sub-analysis, some of the clinical studies, like we published a study on using, we actually use hydrogen producing tablets, and it was 6 months in metabolic syndrome, and we found improvements in cholesterol and glucose and and liver enzymes and weight loss and things, but looking at the data, it almost seems that some people are going to respond better than other people do, for example. And so trying to hone in on what this sensitivity might be. And it's possible that maybe it has something to do with our gut microbiome, because we naturally produce hydrogen gas in our intestines, right? Produce hydrogen gas naturally. But people produce different levels. Some people produce a lot. Some people don't produce any at all, in fact. And then it's going to depend on our diet, what type of diets we're going to have. And so there might be something going on here that would make one person more sensitive to another, and hopefully we can hone in on that. And then we were talking earlier about I did update our website, molecularhydrogeninstitute.org. So right now we're able to offer some excellent education about molecular hydrogen, what it is and what it isn't. There's so much confusion out there.
Well, you've got some courses too that you're putting together.
Yes. Exactly. Yeah. So I'm hoping that if you're interested, if anyone is interested in learning about molecular hydrogen and just trying to understand the principles and things behind it, so you can answer your own questions, these courses are really going to help, because there's a lot of information out there that's not correct, and so these courses are specifically designed to eradicate a lot of the misinformation, get the correct information, and allow you to think about how to use molecular hydrogen the best, how to optimize, and so on. So I think people are going to really like them.
Perfect. So definitely head over there, molecularhydrogeninstitute.org. Two closing questions. One is the difference, the contraindications. Are there any for molecular hydrogen? I do recall in the past that there was a concern with SIBO, or small intestinal bowel overgrowth. Is that an issue? Or are there any other contraindications that someone should be concerned about using molecular hydrogen?
So we don't know. That's just a possibility, right? Because if you have, and there's different types of SIBO, right, where you have the methanogenesis and more, or just a hydrogen producing. And so if they're using a lot of molecular hydrogen as as an energy source, the bacteria, to produce more methane, for example, then maybe if you're feeding more of that hydrogen, then it could be problematic. That could be true for some people, but for other people, it probably wouldn't be true because they also have a lot of hydrogen-producing bacteria. So there's already a lot of substrate in the intestines themselves. And so adding a little bit more molecular hydrogen may not change anything at all. In fact, maybe there's even a negative feedback, process that's going on that can make it better. So if that's a concern, I mean, you could try it, and if you feel worse or something, then maybe it's not for you right now. So there's that aspect, and then in terms of contraindications, again, currently with what we have seen in the literature, there does not appear to be any contraindications. The bigger contraindications would be if you are using your method of making or using a molecular hydrogen is not a very good way, for example. So, sometimes I've seen people take just pieces of metal and put it in water, and then they'll do electrolysis because they'll produce a little bit of hydrogen gas in there. Yeah, you do make some hydrogen, water, but the concentration is low, and then all that electrode material can be degraded and and then leach into the water, basically, and then you're drinking that. So that of course could make you not feel very good or make you sick or cause problems, or if you're overdosing on I don't know, drinking too much water, right? Drinking several gallons of water a day of hydrogen water or something, trying to get a bunch. Well, that's just too much water. It's going to mess up your electrolyte balance. So, but in terms of just molecular hydrogen, still there doesn't appear to be any contraindications. So it seems to be something very safe, and honestly, that's why I feel comfortable talking about it, even recommending it. At least I certainly don't discourage people from using molecular hydrogen. It's just that the safety profile of molecular hydrogen is very high. We see this from many cell culture and animal studies and human clinical studies, and it's naturally produced in our intestines and everything else. And so because the safety profile is so high and the potential benefits appear to be quite remarkable, then the risk to benefit ratio is really quite favorable, and so I think we can go ahead and try to enjoy the benefits of molecular hydrogen without.
Forget to try; you can just enjoy them.
Yeah,
you can do it. It's really great. So finally, with respect to alternative methods of administration. What about, can you review hydrogen gas? Because there's some devices out there that make hydrogen gas. I've been in the process of seeking to get one made overseas, and I'm seeking to get one made for you too, but like a 7% concentration. Totally different mechanisms. Can you just maybe provide some insights as to the comparisons of the two? Now, the gas machines are and you could actually use hydrogen gas itself, like from a same company that would sell you oxygen gas. You can breathe that in. So but that would be, I think, 100%. Maybe you can combine it with oxygen.
Right. Yeah, we do that in the laboratory, and in fact, this is the concern that. Okay, in most clinical studies, that's exactly, maybe not most anymore, but in many of the clinical studies, especially the ones that are done in the hospital settings, that's what it is done. Is it's a tank of very pure hydrogen gas that is mixed with medical grade, oxygen gas, and so it's just very high purity, right? And so the percentages are exact. And so when you inhale that, you're inhaling that specific percentage of 2%, 4%, or whatever it is. Now, when it comes to these machines that produce the molecular hydrogen, that's all great, assuming that the machine is not, putting contaminants, into the gas or something like this. Normally shouldn't be a problem, but the issue is the volume of hydrogen gas even going to be enough? Because just because something is 2% or 1% or 7% or something, well, 7% of 10 is 7. 7% of 10 milliliters per minute is 7 milliliters per minute, and 7 milliliters per minute is not going to be therapeutic for you. So you have to make sure the volume is much higher, closer to 200 to 300 milliliters per minute, kind of at a minimum, based upon the studies. So there's a number of things to look at in this, but then in terms of the benefits, say compared to hydrogen water, more research needs to be done on that. And in some cases, it appears that the drinking hydrogen water can be more effective. And in fact, in certain cases, it's like 100 times more effective. Just looking at their different protein expressions, for example. But in other scenarios, the inhalation might be able to work on different pathways, different areas that the drinking cannot. And so they don't really compete with each other, and there could be an additive or synergistic benefit from them. In the past, I would say most of the research is done on drinking hydrogen water, and that's in clinical studies, and that's still probably the case. But are more and more clinical studies being done now with the inhalation of molecular hydrogen that are showing favorable effects.
Okay, that's great. All right. Well, thank you for that. I think we're going, because I had these technical difficulties with my video camera, I had to switch to backup. I think we're going to probably end it there. But I really appreciate everything you're doing and all the information you shared with us. And thanks for everything that you put together so far.
Yeah, my pleasure. Always great to talk about hydrogen, and thank you for your time, and I appreciate it.
Thanks so much for watching. Remember, hit the like and subscribe button so you can get more videos that can help you and your family take control of your health.